welcome everybody to this H3D Foundation webinar. I'm Nicola Elliott-Wong, and it's my great pleasure and honor to host these sessions. Oh, Franco, we're actually seeing presenter mode. We're seeing your movements. Yeah, we can get it back. That's perfect. Okay, today we are really blessed to have a great friend of H3D, a great friend of the H3D Foundation with us, who's been a friend for a long time. And I know many of you know him, but nonetheless, I'm going to do the introduction as per usual, so bear with me. So Dr. Franco Lombardo holds an MS in Pharmaceutical Chemistry and Technology from Sapienza, University of Rome, and a Ph.D. in Organic Chemistry from the University of Minnesota. He brings over 30 years of experience in the pharmaceutical industry, having held positions at leading organizations, including Alkermes, Biogen, Novartis, Pfizer, and Merck Research Laboratories. Dr. Lombardo's primary interests and area of expertise reside in ADME, physicochemical properties, determination and modeling, in vitro, in vivo, and in silico. He has co-authored over 50 research publications and book chapters, as well as presented invited lectures and seminars at universities and industrial conferences worldwide. He is a co-author of the work on the Rule of Five, ADDR 1997. Following his industry career, he started his ADME PK consulting firm, CMAX DMPK LLC, which provides expertise and consulting on physicochemical properties of drugs, ADME PK, and human dose prediction, together with IND writing assistance. He holds an affiliate research professorship appointment at Northeastern University, Department of Chemistry and Chemical Biology in Boston, and an adjunct professorship in the Department of Medicinal Chemistry at the University of Minnesota. He has taught short courses in ADME at several universities, such as the University of Cape Town, the University of Rome, Northeastern University, and the University of Minnesota. Since August 2025, he's been a member of the Scientific and Technical Advisory Committee of the African Institute for Biomedical Science and Technology in Harare, Zimbabwe. And I should also mention he's been a mentor in our Global Health Mentorship Project for several cohorts. So it's a great pleasure and honor to have Franco with us today. Thank you so much for doing this and for giving us your whys and hows of pharmacokinetics and drug discovery. So everybody, welcome to the session. There's going to be a Q&A at the end. Please pop your questions in the chat or write them down. I'll unmute you later. And I'm going to hand over to you, Franco. So please enjoy, and thank you for being with us.
Thank you. Thank you for the very kind presentation, and welcome everyone. There is quite a bit of material, so we want to get going. And I did add a few slides after the end so that you have additional material and references, which I try to give you as detailed as possible. A basic definition to get into the subject is the pharmacokinetics is essentially the time course of a drug or xenobiotic in general in the body. It is what the body does to the drug, while pharmacodynamics, again, but in a very simple manner, is what the drug does to the body. It is largely determined by the physicochemical properties of the drug in the interaction drug-body. And generally, the way we look at is their physicochemical properties underlie ADME, absorption, distribution, metabolism, excretion, and that yields the pharmacokinetics behavior of the drug. Physicochemical properties, again, ADME plus safety and potency, which is important, will lead us to the pharmacokinetics. A relatively recent, and this is not an easy job, as you well know, because even a relatively recent paper by this group says that delivery of quality candidate drugs requires iterative optimization of physicochemical properties, and we need to make sure that a potent drug in vitro get to the site of action and a clinical hypothesis can be tested. With thanks to the maker of this cartoon, A drug is not something in a Eppendorf tube or in a well. There is a potent compound in vitro, but it's not a drug. A drug is something that reaches the target and does its job. And there seems very little space between those two pictures, but there is a long, long way to go and a lot of money involved in that transition. This is not to say that affinity, this is a very recent, this year paper by Mark Murkoff, this is not to say that affinity is not important. Affinity is important, and you see a paper by about 35 years ago by Ralph Ishman, it still was saying what the same thing that Santesteban 2024 was saying, that discovering an active compound is relatively easy, but discovering an important new drug remains unbelievably difficult. This is true today. With all our knowledge accumulated, with all our tools, with everything. What is the definition of drug-like? Some people say that such definition is defunct. There is no such thing as drug-like. Well, if you look at structure, perhaps no, because you could have azithromycin, which is an antibiotic for upper respiratory tract infections, which is a macrolide, and ibuprofen, which is a non-steroidal anti-inflammatory. They couldn't be more diverse structurally, but they are drugs, nevertheless. So pharmacokinetic friendly could be one definition of drug-like, because we have to deliver this drug from a Eppendorf tube, a Petri dish, or whatever you prefer, or well, into a patient, and so that in such a way that it's safe and it's also efficacious. So Zhao concludes that we need to read, reason about pharmacokinetics. Why do we worry? Well, if we have good pharmacokinetic, if we have convenient dosing regimens, we don't have to take the drug 4 times a day. Sometimes it happens, and then there is a higher patient compliance, and there is better efficacy. And this better efficacy also will lead to a competitive advantage. And for example, you may not be first in class or the first in the market, but you may be best in class. So you may recover the ground that you have lost because you have a better efficacy drug. And obviously, convenient dosing regimen involves the vast majority of drugs we want to be oral, because it's much easier, it's cheaper, doesn't have to be sterile, and who wants to poke an arm or a thigh or whatever with a needle when we can take something orally. Not always this is possible, but we try to strive to do that. How do we study pharmacokinetics? Well, there is an array of pharmacokinetic models. There is a non-compartmental. In the simplest form, we measure the plasma level and we calculate the various parameters. I'll show you what they mean. Then we have the compartmental. We assume that the body is not one single big compartment, but we have 1, 2, or 3 compartments in general. More than 3, we should look at the data, because it is usually usually between 1 and 2. Most drugs show a 2-compartment behavior. Sometimes you need 3 compartments. 4 compartment would be there is something odd about the behavior of that drug. I never really seen used, and even 3 is fairly rare. And then there is physiologically based PK. Physiologically based PK has a lot of advantages, but some disadvantages too. It tries to dissect the body in many compartments. It looks at the cues. There are the blood flow, both venous and arterial. there are the amount of enzymes, the amount of transporters, et cetera, in each tissues that they, especially the developer, you can develop your own, but there are hundreds of differential equations. So more often than not, people use stripped down version if they develop their own. The commercial packages are much more sophisticated, but are much more expensive, as you may imagine. So that's what it does. You can do a lot of simulations with that. If you want a condensed review of PK and PD, is almost a mini book, is this 27, 28 pages, but there is all the terminology, there are examples and so on and so forth. I strongly recommend this work, this pharmacokinetics sort of review compendium that was published a few years back. For a review of PK/PD, which is the relationship between R concentration and effect, in a very, very quick and simple manner. You can take a look at the work by Jusko in Journal of Pharmaceutical Sciences. And there are also different compartmental model, and you can look at these two papers in which there is also the example, as I was telling you, some people do their own as minimal PBPK model instead of having a super sophisticated with sort of all the compartment in the body. Non-compartmental analysis generally is done by taking samples, that's C0, C1, C2. By the way, the C0 you can't really measure. It's an extrapolation from a log linear regression to C0 from C1 and C2, because no matter how fast you go, when you go to the contralateral arm, the other arm from you have injected in one and you sample it from the other, it's at least minimum 3 minutes. So however small that may seem to you, you do lose the trapezoid C0, C1 if you didn't do that extrapolation. So you can do a linear or a logarithm trapezoidal rule. And in general, in general, I do generally linear, but some people recommend to do linear up, log down, because there could be underestimation, overestimation in the ascending or descending portion, depending especially how often you're taking your samples. Instead, the typical plot for an extravascular dose, as you see, there is an absorption phase, and then there is a equilibrium between absorption and elimination at the Cmax at the very top, and then the last few points. Generally, we use 3 in really normal way of doing this in general. And we can calculate the KEL, which is the negative slope of the terminal phase. So in this case, we do this to estimate what we have measured. So from C6 to infinity, because you see we are close to 0, but not 0. This is, of course, an example. We need to estimate this kind of triangles to infinity. So here we would take C6 divided by KEL, and this will give us the AUC from C last to infinity. And in general, this should not be higher than 20% of the total, because if it is, that means that we have not followed the half-life long enough, and C6 in this particular case would be much higher than where it is. So we haven't really waited for most of the drug to be eliminated from the body. Generally, we use this kind of empirical rule, but it is reasonably well accepted. And if the T1/2, the half-life of the terminal phase, is very long, that means we have to sample for very long time. compartmental pharmacokinetic analysis. In this particular case, we look at, in this case, there is a two compartment. We look also at the distribution. So we have an IV injection just before in the compartment one, which is the vasculature, the systemic or central, they're kind of synonym. And then there is the distribution going into tissues in general, compartment two. It's a compartment that is not the blood, okay? So we don't define specifically what it is. And then it comes back and it's eliminated, the assumption it is eliminated from the blood or plasma, whatever we have sampled. How do we distinguish between one and two compartments? When we plot, and only when we plot the scale, the logarithmic scale, the concentration, we can tell if it is a straight line, that's one compartment. If it is a line with a curvature with two faces, sometimes there can be even three faces with different slopes of this, then is a two compartment. Again, I cite Fan and Delanoy, which I very much recommended. We can do these calculations by fitting all these points with nonlinear fit, or we can do what is shown there, a curved stripping. So essentially, I take the slope of the last 3 points, and I calculate the intercept to the concentration, which is B, and then I take each point from that curving portion, and I subtract the theoretical point that you would have on the B line from that. Since it's logarithmic, the difference would be closer to the upper point than to the hypothetical point on the B line, and I form what is the slope A, and that's the distribution. So the negative slope is the alpha, the distribution rate constant, and the negative slope of the terminal phase is beta, as I showed you. The 2.303 is, of course, because I'm using here the decimal logarithm as opposed to the natural logarithm. And that's how we can do the calculations. Then we can calculate the area under the curve. We can calculate a series of constants. We go to the A of ADME and we talk to the absorption bioavailability. Now we try to dissect the ADME. Absorption and bioavailability are not interchangeable. Bioavailability is a much more complex process. Absorption is the first step, if you will, and absorption, you may think also that the absorption from intramuscular, from subcutaneous, not just oral, is whenever there is, and they may be very, very fast, but whenever there is no direct injection in the blood, intravenous. So the bioavailability instead is defined as the area under the curve from route 1 divided by the area under the curve with route 2, corrected, it's cross multiply, corrected by the dose. If the dose is the same, of course, that second term goes away, would be 1. If the route 2 is intravenous, we have given, this is completely bioavailable, of course, then it's called absolute. So I gave a tablet, I compare the AUC of the tablet route 1 to the AUC obtained, let's say by the same dose for simplicity, by giving it intravascularly, and that's route 2. The ratio of the two, either times 100 or not, depends how you want to report it, is your absolute bioavailability. If instead, maybe you're doing a study of bioequivalence and you want to know that formulation A and formulation B are very similar, so they're bioequivalent, or a solution and a tablet are very similar, then AUC2 can be your reference formulation, is still given orally or intramuscularly or whatever, and the other one would be still route 1, maybe a tablet is given orally or intramuscularly. Now here, the route is the same. You could also do it that they're not the same, but let's not complicate things. Here, the route is the same. So I gave a solution as a route 2, and that's my reference, and oral. And then I give a tablet as route 1. This dose is the same. And here is the relative, because it's relative not to the 100% bioavailable IV, but is relative to the performance, the AUC of another formulation. F is generally considered the product, and for the chemists among you, it's like a yield. So the fraction absorbed, whatever is that is absorbed and gets into the enterocyte, fraction that escapes the gut metabolism. Yes, the enterocytes have the possibility of metabolizing drugs. And then from the portal vein into the liver, whatever escapes the liver and goes into the systemic circulation. Now, if everything is 1, you have 100% FA, 100% escapes, there is no gut metabolism, there is no liver metabolism, your F is 1, obviously. And the bioavailability also could be influenced by food, either negatively, you could have a Cmax, so the maximum point, and the AUC, the area under the curve, with food can come down, either or both, or can go up. I apologize that the arrows are inverted. That's my mistake. I meant to say positively it goes up, obviously, AUC, and negatively goes down. But maybe you remember better now, if you think so. That's reverse. My apology. So negatively, they either or both go down, Cmax or AUC. Positively, you have a boosting of Cmax or AUC. Generally, a single species. How do we study before getting to man? Well, we study in animal. Generally, there is no single species can predict, maybe for compound A, yes, but then for compound B, no. So, and then there are particle size, or we give species-specific factor, etc., and formulation. We can change dramatically from one formulation to another. However, we consider an average of multiple species as a reasonable approximation. If you look at the paper by Master, you see that perhaps monkey show a kind of diffuse but a trend. The other dog, rat, and mouse look like a scattergram. Maybe compound to compound, yes, but the other, in general, no. An important construct which is useful in development because a class 1 can get a bio waiver. In other words, if I want to sell my generic and I can demonstrate that my drug is a high solubility, high permeability, and rapid dissolution, those are the drug we love to have, but unfortunately, more often than not, we do not. Then on the basis of in vitro data, I don't need to do a bioequivalent study, which I just described to you. So I would give my tablet, the tablet of the competitor, and within certain limits, I say that my F is the same, okay? And that's not like a full clinical study for a new drug, but it's still expensive. So this one was developed because of that, and the FDA accepts that. In general, in discovery, we look at these compounds when we say, okay, it's fairly low solubility. The 250 milliliter threshold is not, this is something that has to do more with the development. It is the amount of liquid we give to a subject when we do first clinical studies. It's certainly not what's in the intestine. We exchange liters of water during the day, but that's the 8 ounce is what generally is given. So that's why it is considered that. And then the permeability is determined in KCO2 or MDCK system. These are monolayer cells that are immortalized and very, very important to do permeability studies. And we look at the about 5 times 10 to the minus 6 centimeter per second. That's considered a reasonable threshold, but you can find in the literature that the people vary that sometimes, with beware of the assumption. Here is MAD. MAD is a useful construct, and to show you what really matters, its solubility, those are the variables, and Ka. Ka is the absorption rate constant that can be derived from in vitro measurement, from in vivo measurement in animals, etc., etc. The solubility, I do show 6.5, but in general, we take 6.8. That's the original formulation of the equation. 6.8 is some kind of average of the pH where most of the absorption takes place, which is jejunum and ileum. That's why the small intestine is very long, has an enormous surface area. You guys know villi, microvilli, enterocytes, etc., etc. And that's why the role is to absorb nutrients and drugs. The absorption rate constant can be done from P effective, which will be what I just showed you, transformed from KCO2 and MDCK, transforming a human equivalent through these empirical equations. And then Ka, you see on the right, where R is the diameter, the radius of small intestine, and you can have a Ka. If you really have no idea of your Ka is, if you use about 1.2, you'll be okay. So as long as you've measured the solubility at pH 6.8, hopefully thermodynamic from a crystalline material, and you can do this calculation. Intestinal fluid volume, remember the 250 ml, obviously is a figure of merit. 270 minutes or 4.5 hours as the small intestine transit time. That's what SITT means. And you do the calculation and you get MAD in milligrams. If your MAD is much, much lower than the efficacious dose that you think you're going to need for efficacy, then you may want to start asking some questions to show you the solubility and permeability are important. This is absorption, not bioavailability. Remember, you still have, after absorption, you have the FG and FAH, but is an important parameter and was developed by Kevin Johnson of Pfizer at the time, 96, Becky Hines, etc. And you have also the references where I got those equations from. So those are empirical and they're not universal, but they work reasonably well. And again, if you are lost in the woods and you don't know what your Ka is, 1 to 1.2 reciprocal hour is probably an okay approximation average. PBPK approach. This is a very complex construct, and there are PK-Sim, I believe, is still freely available, but there is GastroPlus and SimSip, and they're fairly expensive. SimSip is really super expensive, but has gained a lot of momentum, especially with FDA. What happens here is that while the other models, the non-compartmental and compartmental, I am treating it as an average animal, average human. Here, you can really modulate what happens, for example, in pregnancy, what happens if you have renal impairment in subject. I can turn off, I use this some of the knobs and cursors of this complex and say, well, my subject has a very low renal function, and if my compound is eliminated primarily renally, chances are I'm going to see an increase in AUC because the compound is not eliminated. Vice versa, if it's the human system that I'm using, the human is not renally impaired, then there should not be an impact. Same thing, hepatic impairment. I can look at genetics. I can look at race. For example, some Caucasians don't have 10% to 6 to 10% of them don't have 2D6 or they're poor metabolizer of cytochrome P450 2D6, which is a powerful oxidative system in the liver that metabolizes xenobiotics, but also some endogenous compound. So if I do have that and my compound is metabolized in a large part by 2D6, I may have an adjustment of dose to be made. Otherwise, I may get an excessive concentration and potentially side effects. I can look at dissolution rate. I can look at particle size. So I can really modify and play with a lot of these knobs and cursors. However, you have to be careful because obviously you have to do things to test certain hypotheses that have certain foundation in reality, because you can make it by manipulating whatever you want. You can make any PK profile look the way you want. The Miller kind of warns us and says the complexity of PBPK model, in which include many adjustable parameters, mandates a definition of consistent model building strategy and best practice guidance. Volume distribution. This is something that is used in combination with clearance, which we'll define in a minute. There is no such thing as a good or bad. It's just the distribution in the body. We don't know where it's going, but it's leaving the central compartment, the blood. The observed range goes from 0.03 liter per kilogram, so just about plasma amount, and to 700 liter per kilogram. You realize that there is no such thing as an individual that has 700 liter per kilogram. This is due to the fact that that's a basic lipophilic amine and sticks to the phospholipids everywhere. So it looks like it diffuses everywhere. We don't know where. This is sort of a weighted average. We don't know where. It's not that it's one compartment to the other just by measuring the log D. We know that has left the vasculature. The most important parameter, and I try to accelerate a little bit, time is a tyrant, and most important parameter is clearance. The total clearance is usually measured IV. You see a two compartment. We give a dose and we measure the AUC, the area under the curve. The dose divided area under the curve is the total clearance. The clearance is additive. Those are the principles I want you to keep in mind. So there is a clearance that is metabolic, clearance that is renal, clearance that is biliary. And it's independent from the fluid where it was measured. Some diagnostic are possible. For example, if I see a clearance that is much, much higher than 20.7 milliliter per minute per kilogram, which is the hepatic blood flow, well, obviously something else is also helping the clearance, because the liver cannot metabolize something at a rate faster than it receives it. And most of the metabolic work you see up there is done by the liver. Cytochrome P450, aldehyde oxidase, FMOs, flavin monooxidases, uridyl glucuronil transferases, glutathione transferases, etc. But there are other clearing organs. An important construct, which is fairly simple to keep in mind, is the extended clearance classification system. Here, we're not giving you the rate. They're not giving you but they're giving you a likelihood of a mechanism of clearance. So if we have high permeable compound, low permeable compound, you see things come together now because you have the KCO2 of the MDCK, and whether the acid zwitterion or base is neutral plus the molecular weight. So they are relatively simple parameters to figure out. And for example, a class 1A, which is an acid of zwitterion with a molecular weight which is relatively small and it's highly permeable, will be eliminated most likely, there are exceptions, by metabolism, while a low permeable compound that is a base or neutral, most likely is a class 4, will be eliminated by renal clearance. This is a table of scaling because I'll show you in a minute when we do scaling from in vitro to in vivo to predict what the in vivo clearance will be, we do need to use body weight, hepatic blood flow, and so on and so forth. Scaling factor, don't be surprised if you see a wide variety, even across the same group, from one publication to the next, you can see different adjustment. I don't know the basis of what, but that's what you can see. The important thing is your own work, you adopt a certain consistent approach. Hepatic clearance cannot be greater than the hepatic blood flow, I told you already. And in general, hepatic clearance is 70% of the blood flow is considered high, in between moderate and below. It's 25%, below 25% is considered low. Here is how we do it. So I represented a petri dish, but of course you done in well and other things. It's either through cells, hepatocytes, or subcellular organelles like microsomes, or even cytosol, etc. Usually from hepatocytes, but you can do also that from intestine. I told you that enterocytes have a repertoire of metabolizing enzyme. The equation that you see there, first intimidating, is nothing more than the Well-Steer model that says, well, the fraction unbound in blood, the clear and intrinsic that is scaled by certain parameter from what we measured with the disappearance in vitro of your parent compound, and then the fraction unbound in microsome or in hepatocytes, because you want to measure that too, because if they bind unspecifically even to the plastic or somewhere else, they won't be available, obviously, to be metabolized. The RB is concentration blood to plasma. In general, if it's 1, we can use that equation interchangeably with blood to plasma. More often than not it is, but it can be a little bit different. And so the clearance in blood, the clearance in plasma change, and the clearance in plasma will be higher if, of course, we partition the compound in erythrocytes, because we eliminate that by spinning down the blood, and it's like the blood, like the drug has been metabolized. And it's still there, except that we take a picture by mass spec of plasma, and it's not in plasma, so it seems that it has disappeared, and your clearance would be much higher because your AUC would be much smaller. Those papers, relatively recent, especially Gardner, show you the comparison and the fact that in some cases you can use surrogate one tissue to another or one species to another. One paper I recommend, although a bit dated, but is still a very good one, is an example of a very, very accurate work done on 29 drugs and shows you different models. There's Well-Steer and there is the parallel tube. And you see the use of fraction unbound in microsome, fraction unbound in blood. There is a variety of recipes where people have used this scaling factor, that scaling factor. There is also they saying that they use scaling factor based on the intrinsic clearance. So the higher the intrinsic clearance, the lower the scaling factor. I'm not sure I buy all of this. I'm not sure I buy the fact that we, in general, you read in the literature that we underestimate the in vivo by using in vitro. If you do, with the necessary check and balance, I'm not sure that's the case. That brings us to the issues that sometimes we find that microsomes, which should be subcellular organelles, and they should be less powerful in terms of metabolizing power than hepatocytes. Hepatocytes contain cytosolic, contain enzymes, contain membrane bound, and contain microsomes, etc., etc. And should the latter be better? Well, some people started to look at that and concluded that maybe the permeability of suspended hepatocyte, this is not a liver, this is a frozen and then thawed suspended hepatocyte, maybe by not being in the microenvironment with the other cells that compose the liver and the vasculature and the various compounds that hormonal-like effect and so on and so forth. Maybe there is a permeability, so the compound doesn't go in. If it went in, it would be metabolized, and if we offer instead to the microsome, that will do a higher, they will show higher metabolism. Then other people said, no, wait a minute. It's not so much. It goes in, but hepatocytes and S9. S9 is microsome plus the cytosol, so there is no longer the hepatocyte intact. There is no longer the membrane, okay? And there are better systems. So this one is only 2 compounds, but done quite a lot vertically to analyze a lot of issues. And maybe there is the competition between binding and even protein-protein interaction between cytochrome P450 and this fatty acid protein binding. And then, again, somebody else also looks at the redox partner. There's no time to go into the mechanism, but remember, this is a redox system, and they say that maybe there is a mismatch in the enzyme redox partnership that is behind this human hepatocyte lysate versus intact human hepatocytes. So that those are the things that you may ask yourself when, for example, assuming that you did both, because if you didn't do clearance in hepatocytes and microsome, you will never know that that you have the discrepancy. Of course, for you, whatever you observe in one system or the other will be the result. Williamson also discusses some of these issues. Plasma protein binding. This is, we're trying to come to an end to give you some time for questions. Plasma protein binding is not a key parameter to modulate. Don't be fooled by it. It's neutral. It's neutral on free concentration. It's neutral on half-life. Neutral on dose. So you will not. It's important to measure, because when we do PK/PD, we want to know what our free concentration is. We can make importance in PK and dose prediction, but do not waste your time in optimizing discovery fraction unbound in plasma, FU or FUP, because there are drugs that have abysmal 0.0001 or whatever and there are drugs. And there are drugs that have one, so they're completely free. They don't bind to plasma proteins, and they're drugs as well. Important transporters, again, those the colors are because they reflect the category. So when it's really confirmed and important for xenobiotics, is red. B, there is some importance, and then C, a little weak, the involvement with xenobiotics. But you see, they are everywhere in the blood-brain barrier. Below on the left in the placenta, and they're a protection. Sometimes also they take in nutrients in the brain. If you didn't have a glucose transporter, the brain wouldn't work because the glucose will never go through the blood-brain barrier as polar as it is. So these are very, very important. Transporter-based drug-drug interaction. This is something that can happen both with enzymes and with transporter. Suppose I'll stick with the yellow and red section. Suppose that you take a nutraceutical or another drug, maybe another drug, you tell the doctor, but you take hypericum perforatum, which is St. John's wort, considered an antidepressant, is an inducer of cytochrome P450, 3A4, and PGP. Well, cytochrome P450, 3A4, and PGP, which is a transporter, P-glycoprotein, cytochrome P450, 3A4 is also highly represented in the intestine and in the liver, does the lion's share of metabolism among the cytochrome P450. If you induce its production, then you're going to have more. And if your drug is metabolized at a higher level, you may not have a therapeutic effect. The reverse is true. If you take bergamottin, naringenin, or flavonoids in this Jabara juice, they're all inhibitor. Now I'm inhibiting 3A4 to D6. If my compound is metabolized by a large extent by 3A4, and you are inhibiting 3A4, now you can have too much of the compound. And if your therapeutic index, the ratio between toxic concentration and therapeutic concentration, is not very large, so the drug is not very manageable, you may be in serious trouble or side effect, even fatal. So that's important. And you don't tell the doctor that you take hypericum. Heck, it's a herbal remedy, right? I mean, what can be the harm? Well, there are. A quick overview of renal excretion. Renal excretion is we won't go through these exercises, but are there for you if you want to look at them. Essentially is glomerular filtration of the free drug. You see there is a few glomerular filtration, and the tubular secretion, because there is a secretionary and tubular reabsorption. The first two eliminate drug. The third term, of course, is negative because it tends to reabsorb within the blood from the tubular lumen. The ultimate top, the condensation of what we've been talking about is to predict human dose and dosing regimen. There are a variety of approach to do that, and you can see in the paper is a nice review approach with first in human. That's what FIH means. Human parameters can be predicted with a variety of approaches. Human parameter and pharmacologically relevant concentration, target engagement, target suppression is likely to yield a beneficial therapeutic effect. But I have to have a solid biology and pharmacology. I have to know that, yes, if I have enough drug at the receptor site and engages, most likely I will have my pharmacodynamic effects and therefore my therapeutic benefit. We make assumption on linearity, we make assumption very important. We make assumption, for example, that the cancer, even though it's from a human, patient-derived xenograft, PDX, is implanted on the flank of a mouse, is really resemblant of the human tumor that grows, for example, in the colon in a human. Well, that's not really the case, or may not be the case. We do shrinkage of the tumor, and we probably progress the compound. However, most of the time, we lose compounds because either we miss one toxicity in animals that we couldn't see, that can be idiosyncratic no matter how well we do it. B, because our model of the disease, however well tested, etc., may not be representative of the human disease. And therefore, even the shrinkage of that tumor does not correspond to a breast cancer treatment that will work in human, or a colon cancer, etc. This is essentially to show you, and this is our last slide, that why do we do what we do? Well, volume of distribution and clearance tell us something about half-life, how long the compound lasts, and therefore how often, once a day, twice a day, 3 times a day. Clearance and absorption, bioavailability, I told you absorption and bioavailability are not synonyms. How much? So if I have a poor bioavailability, I will likely have to increase the dose. These two equations are very empirical with some of the parameters you can guess. In particular, this one is accurate within a factor of 10, which is not exactly something to write home about, because between 3 and 30, I may say, well, it's going to be a low dose. Okay, but if it's 300 and 3,000, you realize things change. And if I'm accurate within 300 to 3,000, I'm not sure that, I want to use this equation. And as I promised, there was the one a little bit later than expected, but I'm at your disposal if you want to engage in some questions.
Fantastic. Thank you so much. Guys, you have been unmuted, or at least there's an option there. So please raise your hand if you have any questions. And Franco, if you're up to it, you can stop sharing. And that might be easier to see. Or if you want to keep your slides online, that's also fine. Yeah, all the applause coming through. So there was one question in the Q&A section, which I will read to kick us off, but then please do raise your hand to get in line. I'm curious, says Michael Rabella, can the elimination rate constant be calculated using the same in concentration versus time slope for both IV and oral administration, or are there differences in how we should handle the oral data?
You can calculate, if I understood the question correctly, the question is if we can calculate absorption rate constant.
He's going to have to ask the question himself. Michael, do you want to jump ahead if you're still online? Otherwise, Matthew, you might be able to explain the go ahead. Matthew, if you can help us with this question.
No, I wanted to ask a different question.
Yes. Okay. Go ahead with your question, and Michael, please jump in to help us if you're still around.
Sure. Thanks so much, Franco. I think this has been, as usual, a really good talk, and lots of things for us to think about as we continue to progress drugs through the discovery cascade. I want to think a little bit more about the point you made around the different explanations for the disconnect between microsomes and hepatocytes. And of course, in a number of the papers that where people have done this analysis, people have the benefit of kind of working back from clinical data to say, if we have this information, then this is what would have chosen.
Yes.
in points like in early discovery where we are, where you only have essentially microsome and hepatocyte data, maybe you have some in vivo data points as well, and you don't know what the clinical side is going to look like, and microsomes say one thing, hepatocytes say another thing. Is there a way to kind of, I don't know, is there more experiments? Is there something you would do in order to just say, if we're going ahead, then we think we'll be closer to the value that we have with microsomes or closer to the value that we have with hepatocytes?
One answer is coming from this paper, which is Tashinga, Bapiro et al. Is in Communications Biology 2025, and it's AstraZeneca group, where they say, well, we think that we should reconsider using microsomes and stick with hepatocytes. That's one way, maybe a bit drastic, I don't know, but they think that that's and those are part of the two references where they did the disconnect with the redox partner mismatch. The other thing is that do you see with the animal data in vivo a certain correspondence or not? Then you may lean toward using the system. There's no proof that it's going to work for human, but you may lean toward using the system that reproduces better the IV clearance behavior of the compound, assuming that your compound is mostly metabolized. Otherwise, there is no reason why you should see a metabolism driven clearance. I'm sorry, there is somebody on there.
So sorry, I'm just trying to find who's causing the feedback. Guys, if you can stay muted until it's your turn to speak. Sorry. Let me just mute everybody and I'll start again.
Can you please mute because thank you. Okay, back to us, Matthew. So I don't see Matthew anymore. But anyway,
I'm still there.
Okay. And then you can do some kind of scaling factor in vitro, in vivo, and try with that to correct for the human. So in other words, if you see that you overestimate or underestimate with microsome or hepatocytes, as I said, my bias is hepatocyte, although it's more expensive, then you can try to correct with that factor your in vitro in human and say, well, maybe the in vivo in human, because if there is a consistent under or overestimation, and a consistent over or underestimation, you may try to cure it that way. That's one way to do it. And most people tend to stick to the hepatocyte. Well, I shouldn't say most. Several people do recommend sticking to the hepatocyte, which is of course more expensive. Some of them could reproduce S9 plus microsome that could reproduce hepatocytes. So that's because also binding to different proteins, chaperone, etc.
Sure.
That's the best I can. Too bad I missed the other questions. If it was a Ka, yes, you can fit the Ka. Ka, there is no there when it's IV because of course there is no absorption rate. You give it IV. But if the question from the other person was if there is a way to calculate, yeah, there is a way to calculate even from oral profile or intramuscular profile. You can do a compartmental fit and you get to the best of your fit AKA, or you can calculate with MRT in IV and PO, or you can calculate with P apparent into P effective into Ka. At least those are the three that I know of. If this person is still online, I don't know. And that was the question.
Thanks so much, Franco.
You're welcome.
Thank you so
Anytime,
much,
guys. I'm an email away, so.
Franco is available. He's been an incredibly active mentor, and yeah, he's always willing to answer kind of any questions we have. So we're always grateful for that. We're at the hour, so I'm going to close the session, but do feel free to send through your questions if necessary, or if you have any burning questions beyond the session. So thanks to all our participants for turning up. Thanks to Matthew for your great question, and a huge thank you to Franco. We're so grateful to have you in our corner, and thank you for your passion and expertise today. We hope to have you back in the future again. So thank you, everybody, and we'll see you next time.